In our third section on this, in this lecture on the profiles of prokaryotic cells, we're going to first take a look at the plasma membrane. And the plasma membrane is mostly a phospholipid bilayer that encloses the cytoplasm, and it's supposed to form a barrier between the water-based interior of the cell and the mostly water-based exterior of the cell. And it's a dual natured molecule. We touched on it in the chemistry lecture. We have the phosphohead groups, and then we have these fatty acid tails that are facing one another, but I'll show you that in a second. And so embedded into this plasma membrane are proteins typically. If they're peripheral, that means they're attached on the surface, either on the outside or on the inside. So they're not spanning the entire phospholipid bilayer. So they're peripheral, they're attached on the outside or on the inside. If we say something is integral, that means it will span the entire membrane. So peripheral versus integral proteins that are attached or associated with the plasma membrane. So here, so after spending lengthy time on the cell wall, so we're now focusing on this phospholipid bilayer. So again, as you know, in gram-positive cells, we have that one phospholipid bilayer, and in gram-negative cells, we have two phospholipid bilayers. So here, this would be, of course, a gram-negative cell where we have an outer membrane and then a lipid bilayer in the inside too. So this is clearly a gram-negative cell wall. It's slightly confusing that the way it's shown here, but so again, the outer membrane, this is here outer layer, outer phospholipid bilayer. Then we have the lipid bilayer of the plasma membrane, so that's the inner one here, the inner layer, and then in between we have that peptidoglycan, so clearly gram-negative cell. So let's move on, and here let's take a look at something called the fluid mosaic model. That means we have these rows of phospholipids. So here would be the polar head groups of all these phospholipids, and then we have the fatty acid tails, and we have two rows of them, and the fatty acid tail layers here, they will face one another. So they have always these fatty acid tails, and they like to face one another to form a nice non-polar barrier to any kind of water leaving the cell. Embedded into the phospholipid bilayer are proteins. Many of them will be receptor proteins, transport proteins. Many of them have form a pore like this one. You can see this right here. This will have a pore that can open or close and let stuff in and out depending on what the cell needs. We have integral membrane proteins that span the entire phospholipid bilayer. So this one is definitely one that happens to also be a channel. This one here has this ribbon structure going through. Those are all integral membrane proteins. This one here as well. And a peripheral membrane protein would be one that's just associated. It's just sort of linked to the phospholipid bilayer. Okay, so the fluid mosaic model is this model of phospholipids, two rows of phospholipids and all the stuff that's embedded in it. A fluid because it literally is a fluid. So the phospholipid bilayer is a liquid. It has approximately the viscosity of olive oil, which is not even that thick, but it's very effective in keeping the water-based interior of the cell inside and not letting water-based stuff from the outside just freely go into the cell. The proteins, they can move freely, and so that means that the whole thing is pretty dynamic. In fact, the proteins, they travel a lot of times in the phospholipid bilayer. It looks like lipid rafts. They're just traveling like as if they're just having a raft, and they're just skating around the surface of the phospholipid bilayer. Also, the phospholipids themselves, they can rotate and move laterally. So going back to this image here one more time. So these phospholipids, these little phosphohead groups right here with the fatty acid tails, they can move around. They can just spin around their own axis. They can also travel. And then these proteins, they're not fixed. They can just kind of zoom around along the phospholipid bilayer. So it's pretty interesting. The whole thing is pretty dynamic, called the fluid mosaic model. And so now the function of this phospholipid bilayer is to provide selective permeability. Again, the cell wants to decide what goes in and out of the cell. And so the phospholipid bilayer makes a great barrier for all the polar and water-based stuff. Also, the phospholipid bilayer contains enzymes for ATP production. Since we don't have mitochondria in prokaryotic cells, we're going to use the phospholipid bilayer of the plasma membrane for ATP production. We'll talk some more about that in another lecture. Some membranes also have photosynthetic pigments that are called chromatophores. So here's some nice pictures here. So here we have some chromatophores that are embedded here into this particular bacterium. And okay, the movement of materials across membranes. So let's spend a little time on that. The phospholipid bilayer is a nice barrier, but we embed typically pores into this phospholipid bilayer so that we can allow the movement of materials across these membranes. Anytime we don't need energy to move stuff across, but we're just relying on concentration gradients, we call that passive processes. Anytime we have a concentration gradient and we open one of these pores, then a substance can go from high to low concentration. So it will go down its concentration gradient. Now, if we don't have a concentration gradient, or we need to actively move something from one place to another, and we just want it to be at a certain another place, then we need to spend energy on that. We grab something, we move it somewhere else, that's going to cost us energy, and so we call that active processes. So a lot of times we establish concentration gradients, a lot of times we go from low concentration to high concentration, but the big thing is we need to put energy into this, and it costs quite a bit of energy in form of ATP. This one over here, no energy spent, which is nice for the passive processes. So let's take a closer look at all the passive processes. First thing comes to mind is simple diffusion. That is just when there's no barrier and a substance moves strictly down a concentration gradient. So we're going from high to low concentration, and this diffusion will continue until there's no more concentration gradient and we have reached what's called an equilibrium. So here an example for that would be, let's say, a gas molecule that can freely diffuse through the phospholipid bilayer because it has no polarity and it's just a small molecule. So simple diffusion means that these molecules that are in high concentration over here on the outside will move down the concentration gradient into the inside of the cell without hindrance until there is no more concentration gradient. That is simple diffusion. It will keep going until there is no more concentration gradient. Now, facilitated diffusion means diffusion with help. So now we need to have help to get across because we are trying to get something across the phospholipid bilayer that wouldn't normally be able to go through that lipid barrier. So that would be something that's charged perhaps, or something that's larger and hydrophilic in nature. For that, we need facilitated diffusion. It needs usually a pore, some sort of a channel or a pore. And that's the way you get ions or larger molecules, especially the hydrophilic ones, down. But you still need to have a concentration gradient. So it's still a passive process and it doesn't require energy, but you need to help the stuff across because the phospholipid bilayer is too good of a barrier. So here is an example of facilitated diffusion. Here we have a non-specific ion channel, apparently because these are charged particles, these are ions, and they need to get across. And here there's a concentration gradient. All we have to do is open this pore and allow those green, whatever they are, ions to go down the concentration gradient. So this would be facilitated diffusion through a non-specific transporter. Now here is a specific transporter. In this case, we're using a glucose transporter. So we have glucose in high concentration outside right here, not much on the inside. So now we need to get the glucose across. It would not be able to cross the phospholipid bilayer, so we need a transporter. We're binding a glucose molecule here to this transporter, and then we're shifting configuration and pushing it out on the other side. Here is our glucose now inside of the cell. A very important passive process is osmosis, which is by definition the water movement across a semi-permeable membrane down a concentration gradient. So we're still we need a concentration gradient for osmosis to happen. In this case, it is the concentration of water that matters. So water will move from the area of high to the area of low concentration and that is called osmosis. Now, when it comes to biological membrane, because water is such a small molecule, a little bit can move through the phospholipid bilayer just by virtue of it being small. And then the majority will have to go through aquaporins, which are water channels. The osmotic pressure is the pressure needed to stop the movement of water across the membrane, so to block it or push against it, against that the osmosis that wants to occur based on concentration gradients. So here is a water channel, typical aquaporin right here. So this would be the preferred way of water getting across a phospholipid bilayer. Keep in mind, water, of course, is polar. It has polarity. And so normally it wouldn't be able to go through the phospholipid bilayer like this. But because it's so small, some of it can go through. But then the majority would go better. It would be better to go through a water channel. Here is a typical kind of osmosis experiment. We have this dialysis tubing, and it's filled with particles. So inside of the bag, there's a lot more dissolved particles, as you can see. That means the water concentration is lower inside of the bag. Let's say we have 10% salt and another 10% sugar in the bag. That leaves only then 80% for the water. Let's say we put this dialysis bag, which is permeable to water. Let's say we put this into pure water. Then on the outside here, we have 100% water, right? H2O, 100%. Now, if we, now in the side of the bag, we have what, 20% dissolved particles, so it leaves us with 80% water. Let me change my color. So let's say we have 80% water here inside of the bag. So now what happens? Water will go from the area of high to the area of low concentration, trying to get it to an equilibrium. So water is going to rush into the bag massively. You can see all these arrows. Water is going to go in, and this bag is going to fill up with more and more water, and all of this is in an attempt to make the water concentration the same inside and outside. Meanwhile, the volume is increasing, and you're pushing up this fluid up this tube, this glass tube, and then you can measure sort of the rate of diffusion of osmosis. And the osmotic pressure will be the pressure that will be required to hold this growing water column or liquid volume down. Okay, so that's osmosis. Now, there are 3 terms that you need to know, and that refers to the tonicity of a solution. Tonicity means strength. And so these are relative terms. An isotonic solution is one where you're comparing solutions. Isotonic means that the 2 solutions I'm comparing to one another have the same amount of dissolved particles. That means the water concentration is also the same. That means there's no net movement of water if I were to place these solutions and separate them by a semi-permeable membrane, if I put them in close proximity. So the solute concentrations are equal inside and outside of, here the examples given of cell. You could also take a dialysis membrane. So that means no net water movement. Water is at equilibrium. If I have a hypotonic solution, hypo means below. That means the 2 solutions that I'm comparing to, the one that's hypotonic has fewer dissolved particles or lower solute concentration than the one I'm comparing to. Let's say we're going back here to our example up here. That would mean that the water here is hypotonic and whatever's in the bag is hypertonic when I'm comparing them to one another. So if I'm comparing the water on the outside to what's in the bag inside, then I would say the water is hypotonic in comparison to what's in the bag. If the bag, what's in there is my reference point, then I would say what's in the bag is hypertonic compared to the water on the outside. So these are relative terms. Hypertonic means that I have a higher concentration of dissolved particles, or higher solute concentration as the solution I'm comparing it to, and here the comparison is the cell. So if I'm placing a cell into a hypertonic solution, that means the solute concentration is higher on the outside of the cell than it is on the inside of the cell. And again, water always goes down a concentration gradient. So if the solute concentration is higher on the outside, than on the inside, that means my water concentration is higher on the inside of the cell. That means water will move out of the cell and the cell will crenate, it will shrivel up. So here are the different scenarios that you should know and be aware of. In isotonic solution, we have the same concentration of dissolved particles inside the cell and outside of the cell. No net movement of water will occur. If I'm placing my cells into a hypotonic solution, now I have a higher concentration of dissolved particles inside of the cell and less water. Therefore, the water concentration is higher on the outside of the cell. So now the water is going to rush into the cell and it may cause lysis. Sometimes it can break the cell. If the cell has a cell wall, it's just going to swell up. But if the cell doesn't have a cell wall, it's just going to burst. Now, our final scenario is going to be the hypertonic solution. I'm not quite sure why it scrolled all the way back down here. Stuff that we already covered, but let me get back to this here. Should be there momentarily. So in a hypertonic solution, you're going to have more dissolved particles on the outside, and I apologize for this. I'm going to try to fix this. I don't know what's going on here. It just keeps going back and forth. I'm going to have to truncate this.